Hello everyone, today we're going to look at genetic mutations and this is for Year 12 GCSE Biology, starting on page 18. If you're doing double award biology, it's starting on page 17. So you've heard the word mutation already in this unit, and we said a mutation was when a mistake happens, whenever things aren't copied properly, when the DNA is being copied or when chromosomes are being copied. If we look at this diagram, we had looked at this before, and we've got the sugars and the phosphates here. That's the backbone of DNA. We just got one strand of the DNA molecule, one strand of the double helix. Along here, we've got different bases. And you know that the different patterns of bases basically represent different amino acids. So we get amino acid 1 there, we're going to get a different there because we've got a different code. And in the last module, we learned that if a mistake happens, so for example, if this G here was changed and became a T, so whenever the DNA was being copied, if that became a T, we'd have ATG. We wouldn't get amino acid 3, but ATG actually would be amino acid 4. So we'd have amino acid 1, 2, 4 and 4. That would give us a different protein because the amino acids would bind in a different way, and therefore you get a different shaped molecule. If it was an enzyme it was coding for, the enzyme would be a different shape, and therefore the active site would be the wrong shape to allow the substrate to fit in, and therefore that protein wouldn't work, and that could have very serious implications. So if we come back here then to the top of the page, if I can manage to do that. Change in the structure of a gene, that's what we were looking at there. Or a change in the number of chromosomes, and we've already looked at Down syndrome. So let's have a look at it in more detail. That's a normal karyotype, of someone who doesn't have a genetic condition. And as you know, they should have 46 chromosomes arranged into 23 pairs, homologous pairs. And you can see they're numbered there, so that's 19, 20, 21, 22, and then this is the 23rd pair. We can see we've got 2 X's, so this is going to be a female. But sometimes mistakes do happen during cell division. And we can see top left is a normal male, top right labelled Edwards syndrome. This happens to be a male, but if we look closely, we can see that instead of having a pair of chromosomes for 18, we've actually got 3. Down here in the bottom left, we have got an extra 13th chromosome, and down here in the bottom right, in the last pair, we've got XXY, which is called Klinefelter syndrome. Now, you don't need to know any of those examples, but you just need to realise that there are conditions where someone might have an extra chromosome. This one, if you look closely at it, hopefully you've spotted that the extra chromosome is here in the 21st pair. And so therefore this is a karyotype from a female with an extra 21st chromosome, and that person then has Down's syndrome. Now, some people call it Down syndrome, some people call it Down. So, let's have a look at this. How might this have happened? Well, normally, a sperm cell, the male, the father has 46 chromosomes, and then meiosis occurs to make sperm cells, and they should have half of the set. And obviously the female starts off with 46 chromosomes, and again we've got meiosis, and you get 4 daughter cells, just like you would get 4 sperm cells and so on, all with 23. So normally this is what happens. One of the sperm cells fertilizes an egg cell, and you get 23 and 23, 46. It doesn't always work like that. Sometimes things do go wrong. So how might that happen? Well, from research, they think that the mistake has happened somewhere in the female, during meiosis in the female's body. So the parent cells start off with 46. Two cells with 23. But over here, they think 24 and 22. One of the pairs of chromosomes didn't separate properly. And they both went to the one side rather than one going to one and one going to the other. So we've got an extra chromosome. Now, if that sperm fertilizes this egg cell with the 24, you're going to get 47 chromosomes. So what you need to do is to copy. You don't need to copy out all this wee bit here. Just need to copy out the numbers there onto page 18. Now, and let's just double check what we've got here. So we have said that what a mutation is could be the change in a gene. And examples of those could be, well, for example, cystic fibrosis. There was a mutation many years ago that has got passed down from generation to generation, and that mutation means the person can't produce certain enzymes, and the condition they have is cystic fibrosis. Haemophilia, colour blindness, Huntington's disease, they're all examples of diseases that are caused by a mutated gene. What can cause mutations? Well, a lot of the time they just happen. In other words, we say that they are spontaneous. In other words, there's no reason or no cause for it. It just suddenly happens out of nowhere. They're spontaneous. But there are some things that can actually increase the chances of a mutation happening. Ionizing radiation, what do we mean by that? We mean ultraviolet light, can damage the genes in your skin cells. Now, normally your skin cells divide because you're losing dead skin cells regularly, and so you've got cells in your skin that are constantly dividing to make new skin. But those cells know when to stop dividing. They have a mechanism that tells them to stop dividing because there's plenty of them. But ultraviolet light seems to damage the gene that controls that, and so therefore these skin cells don't know when to stop, and they keep on dividing. And as you know, uncontrollable cell division is cancer. And so that's one reason for some mutations happening. It could be ultraviolet light. It could be some chemicals, for example, benzene, can cause mutations to happen more regularly than normal. But as we said, many mutations are spontaneous. So in terms of filling this in, meiosis occurs in the testes and ovaries, producing gametes which are haploid. So they have 23 chromosomes. The zygote therefore contains 46. The zygote divides then by mitosis to produce an embryo, and each of the daughter cells, each of the embryo cells contain 46 chromosomes. But we've said sometimes a mutation happens, and it's normally in the mother's body, and so sometimes she will produce an egg with 24 chromosomes, therefore the zygote would end up with 47, and that causes Down syndrome. Some information. Okay, so this graph shows us that, the graph shows us we've got the age of the mother at the bottom here, that it stays pretty, the risk of Down syndrome stays pretty low, but around 35 years, it starts to increase rapidly, the risk of having a child with Down syndrome. And we'll see later on that there are tests that those women can have to see whether they're carrying a child that might have Down syndrome. So So Down syndrome, you need to remember extra chromosome and that it's an extra 21st chromosome. There are other conditions, but this is the only chromosome abnormality that you have to know about. In terms of other mutations, that's a change in the structure of a gene, kind of things like Huntington's disease, cystic fibrosis, haemophilia are examples of genetic conditions due to mutations that have affected the person's health, and they've got a genetic condition. Okay, bye.